Okay, guys, let's start. Assalamu alaikum. So today we have our last lecture for this semester. And we're gonna learn about, so we'll continue on the same kind of path which we were discussing last few lectures about the differentiated state. And then we discussed about the plasticity of differentiated state. And now to take it to a step further, we'll discuss what is regeneration. And regeneration is basically the repair and replacement of missing parts. And the biggest question in regeneration is whether the stem cells are involved in this regeneration process, or it's the adult end cells that de-differentiate and then redifferentiate into the and grow to develop into the missing parts. It is extremely rare in adult animals, but plants can regenerate very well. And if you or add attach a cutting of a plant to another plant, it can grow very well. But in case of animals, it's extremely rare, and especially larger eukaryotes or higher eukaryotes, it's rare. In invertebrates, it's, I would say, more or less frequently seen. In many invertebrates, it's seen that they can regenerate because they are more simpler animals. They have lesser organs or lesser, more simplified body structure, germ layer structure. So they can regenerate their tissues very well. But as you go up in the hierarchy of evolution or hierarchy of the complexity of the animal kingdom, this regenerative ability is completely lost, with one particular exception, or maybe two particular exceptions. So the tailed amphibians, the salamanders, they have this exceptional ability to regenerate their limbs, tails, jaws, even retinas and lens as well. And we also consider this example of adult newt lens regeneration in the last lecture. Where there's pigmented when after lenectomy. So when they surgically remove the lens, the pigmented epithelial cells, they de-differentiated and then redifferentiated into lens making cells. So, and today we're going to cover a little bit more how the salamanders can grow their limbs. So they have retained this remarkable property, which seems to be completely or more or less completely absent in the higher eukaryotes. And if you talk about the tailless amphibians like frogs, they can regenerate up to the larval stages, but after metamorphosis, when they grow into the adult form, they somehow also loses this ability. Another remarkable animal species in the animal kingdom is the zebrafish. So it's a small freshwater fish that is actually commonly found in South Asia. And it is called zebrafish because it has these long stripes, horizontal and posterior stripes. And zebrafish has this remarkable ability to regenerate its heart. So it can regenerate. We will see how it regenerates its heart. But if let's say 15% of its heart ventricle is cut out, it is able to regenerate it back. And when you talk about mammals, mammals can only regenerate the ends of the digits, and except from mice. So mice keep this ability to regenerate their heart for the first 7 days of their life. And we will consider why is that, or how is that possible. But otherwise, generally mammals, the only part that they can regenerate is the end of the digits. So another important point to consider here is that the regenerative ability is not dependent on the age. So, and this was very nicely demonstrated by the experiments in adult newt. So this tailed amphibian, the salamander, type of salamander. What they did was they also have quite, they live quite long. So they removed the lens of these salamanders 18 times, and it was around 30 years of age. And every time the salamander grew it back. So that was a remarkable experiment and remarkable discovery that it's not age dependent. What one might think that this regenerative capability will be lost when the animal ages, but that's not true. At least not in case of salamanders. So yeah, so some of the bigger questions in the regenerative field is that what is the origin of cells that give rise to regenerative structures? And if is it the stem cells or the end cells or the differentiated cells that firstly differentiate and start dividing and growing into the organ that was lost? And then the second most important question is, how is the patterning achieved? Because in case of limb development, so limb has different parts. So you have the most distal part that has your digits, and then you have the more proximal part, which has two bones. It has particular pattern. And then you have on the more proximal, even more proximal part next to the shoulder, you have one bone, at least in mammals. This is how it is. And so there is a pattern to every limb or every organ. For example, if heart is cut out, a heart has a particular shape and topology. And during regeneration, it's very important that the regenerative cells know or have the memory how to pattern that organ when they regenerate and when they repair or replace the missing part by growth of new cells. And so this is a very important consideration during regeneration, that how patterning is achieved. And then the question is it similar to the patterning of structures during embryogenesis? And sometimes it's true and sometimes it's not. So sometimes the patterning is achieved in a completely different way and through different molecular pathways. So we will start the discussion with by seeing how the regeneration occurs in invertebrates. And then we'll go into the amphibian limb development, and then we will also look why mammals cannot regenerate apart from the end of the digits. And then finally, we will look into the zebrafish heart regeneration or in mouse heart regeneration in the first 7 days of their postnatal life. So starting with invertebrates. So there's this invertebrate called Hydra. And Hydra is, what is Hydra? Yeah. So it's a freshwater or a living organism, and it has a typical body structure. It has a body cavity, and then it has this head side and the tail side. And towards the head side, it has a mouth hair, and then it has these tentacles, which are used for approaching the prey or, yeah, for different purposes, for swimming probably as well. So Hydra has this ability to regenerate, and they have a particular type of regenerative capability, and that is known as morpholexis. And what happens in morpholexis is, for example, if you cut this Hydra into two parts at the transverse axis in this gastric region, we will see a little bit in more detail different regions. So in this mid part, transversely cut out, what happens is that both these parts, they grew the part that is missing. So for example, this part is missing the tail, so it grew back the tail, and this part is missing the head, and it grew back the head. But the important thing is that So there is a repatterning of existing tissue. So this tissue was repatterned and it made tail, and the tail part it repatterned and made the head part. And so there is reestablishment of boundaries. But there is the important thing is that in this morpholexis and hydra, there is no cell division and growth. So it's basically, let's say, it's the stretching of the existing tissues that made it possible to repattern the whole animal. And what happens is that the two new animals, they are half the size of the original animal. So then you can understand that since there is no cell division and growth, so each part is basically it grew the lost part, but then the size of the part is half. Size of the whole new animal is half the original one. Then you have the example of starfish, which when you cut out one of its arms, it can regrow, it can grow it back or regenerate it back. And this is known as epimorphosis. And in this case, there is new growth, which leads to patterning of structures. So these are the two kind of examples where lower animals or invertebrates, they replace their missing parts. And this is also kind of explained in this intact flag, French flag system. So it is tricolored. And if you cut it into half at the midpoint, what would happen in morpholexis is that the new positional values are achieved in a way that the whole thing is the size of the half cut. But in case of epimorphosis, the new positional boundaries are achieved by the growth, so that the eventual animal is exactly the same size as the original animal. Okay, so now we consider a little bit in more detail how regeneration in hydra actually occurs. So first we understand that the structure of the hydra, so it has two germ layers, outer epithelium and ectoderm, and inner epithelium, that is endoderm. So there is no mesoderm, so it has only two germ layers. And there are these interstitial adult multipotent stem cells. So we discussed that in hydra, there is no cell division and growth. Yet there are about 25% of the cells of the hydra, they are from the adult multipotent stem cells. And the significance of these cells is not completely understood, but they are there. Yet the hydra, when it is cut into half, it leads to morpholexis. So it regenerates body parts by not growing the cell, but by expanding the body or stretching the body part, the existing body parts, and may replace the missing part. But we will now consider what these adult multipotent stem cells would do. So there is a very high turnover of cells in the gastric region. So this is the gastric region. So we previously saw that if you cut the hydra into half, then there is no cell division that occurs. But if you cut out, for example, part of its mouth or this hypostome region and patch it on the gastric region, on the side of the body, then what would happen is that these interstitial cells or multipotent stem cells, they would multiply and they will grow a new head on the side of this region. So that's the significance of these stem cells. If you cut the whole animal into half, these cells will not activate. But if you cut out part of it and paste it on the side, only then this hydra can grow a new head on this region. And so in hydra, there is this overall polarity. So you have a head region and a tail region. So this is called the anteroposterior polarity. So you have a basal disc at the tail region, and you have a mouth towards the other end, and which also has these tentacles. And this polarity is important because um, the regeneration of lost parts is dependent on this polarity. So this polarity also, so whenever you, for example, this polarity dictates that if you cut the hydra on the face interface, so for example, the cut is made on the face interface, the hydra will always grow a head towards this side. It will never grow a tail on this side. But if you make a cut on the tail side or the basal disc side, if the cut is facing the basal disc side, then it will always grow a basal disc. It will not grow a head on this side. But this was a different experiment because here we take out the cells from this region and put them on the side. And I will explain the significance of this experiment a bit in a minute. So there is this anteroposterior posterior polarity where you have a head region and a tail region, and if there is a cut on the facing the head side, it will always grow a head, and if you make a cut on the basal side, it will always grow a tail or the basal disc. Now, why is that? There must be some signals that determine that this is that the hydra needs to regenerate a head if you make a head facing cut, and there must be some signals that block, for example, head formation if you make a basal facing cut. And this is actually true that the hydra has, for example, two types of signals. So there are these positional signals in the cells. So the inner epithelium and outer epithelium, they somehow they have certain signaling molecules that that determines the head region and the tail region. So for example, in this region, so there is a the inhibitor of head region, or the molecules that inhibit the head, they increase if you go from top to bottom. So that's why if you make a cut here on the basal side, it will never grow into a head because the signal intensity of inhibitory signals for head, they are maximum at the basal side, and vice versa. So if the basal inhibitory signals for basal disc are maximum on this side, so if you make a cut on this side, the inhibition will be very strong and it will never grow into the head region, tail region. So what happens when you take out tissue from here and then put it somewhere towards the basal disc? Then what happens is that the inhibition for head is reduced, and because you are bringing in tissue from a region where the inhibition is very low, and you're putting it in a region where the inhibition was usually high, but then this tissue oversees or supersedes the inhibitory signal, and then the head grows here. So there is anteroposterior polarity, and then there is this organizer, this concept of organizer. That if you, for example, you can induce a secondary axis here, and if you take out cells from here and put them here, it will grow into another head. So yeah, so there is an organization in the body which is dependent on different signaling molecules, which are not completely known, and that's not the topic of discussion here, but it's important to understand that there are positional values that are determined by signaling mechanisms. So there is an inhibition in case of hydra. It's very simple to understand that there is an inhibition of the inhibitory signal for head that increases if you go from head to the tail, And the inhibitory signal for tail, it increases if you go from tail to head. And then, of course, as always, the wind signaling is extremely important, and you will see it in also in other species as well, that wind signaling is very important for body patterning. And if you, for example, just a moment, excuse me. So some experiments have shown, scientists have shown that if you activate the wind signaling in Hydra, you can actually, it can lead to multiple heads production or regeneration of multiple heads. Then we consider another example of this planaria. So planaria are also invertebrates, and it is so yeah, so planaria are these flatworms, and they basically show both morpholexis and epimorphosis. And they have a slightly more complicated structure, but not very complicated. So they do not have any body cavity, but they have 3 germ layers, all the 3 germ layers. But they have a very specific type of cells. These are called neoblasts, and these are kind of stem cells. So they are undifferentiated cells, but they are different from stem cells that they cannot self-renew. But they can be considered as these multipotent progenitors. So they can give rise to different cell types. And there is a heterogeneous population of these cells, which was later found that there are both pluripotent cells and the multipotent cells. And so what they did was, the experiment that demonstrated this pluripotency was they irradiated planaria, and we discussed earlier what happens if you irradiate an animal or cell. So it's dividing cells are completely lost, and the DNA is irreversibly damaged, and the cells cannot divide any further. So all the neoblast cells of that planaria are completely gone. And then they introduced one cell, one neoblast, single neoblast, and some of them, some of these planaria, they and then when they induced a cut in the planaria, some of these planaria grew different organs, but the others did not grow all the different cell types. And this experiment showed us that these there is a heterogeneous population of neoblasts. So some of them are multipotent and some are pluripotent. So as shown here, so if you take out each single cell from these and put it back into the planaria that is irradiated, treated with radiation, so that its own neoblasts are completely gone. So if you put back one by one or one into one planaria, then you can follow what kind of growth it achieves. And this experiment showed that some of the cells, they behaved like pluripotent cells, because they give rise to different differentiated cell types from all the 3 germ layers. But some of them, they gave only very limited kind of lineage, lineage of differentiated cells. So it's a heterogeneous, so neoblasts in planaria, it's a heterogeneous population. So both morpholexis and epimorphosis are involved in planaria regeneration. And neoblasts, if you make a cut, for example, on the head of this flatworm, it grow into a structure, start to grow during regeneration. The first structure that it forms, it's called blastema. So blastema is the basically is a mass of, in this case, it's a mass of these neoblast cells that are formed under the skin. So first there is a covering of epidermal skin that covers the wound. So if you make a wound on the planaria, on the head side, the first thing that happens is that the wound is covered by the epidermal layer, and under that epidermal layer, the blastema is formed, which is basically the growth of neoblast cells. So blastema is very important, and this epidermal apical cap is also very important in limb regeneration, which we will see later in a couple of minutes. So blastema is formed, and yeah, and that is important for regeneration in planaria. And in planaria, there is not only anteroposterior polarity, so it is a little bit more complex than the example of Hydra. So Hydra had only anteroposterior polarity, so from head to tail. But planaria has also, in addition to anteroposterior polarity, it also had dorsoventral polarity, so towards the sides. So it also knows what is the back and what is the front. Also the head and tail, but also the back side. So dorsal side is back, like our back is the dorsal side, and our stomach is, or the chest is the ventral side. So there's also this polarity. So different types of polarity, and it was shown that again the anteroposterior polarity was regulated by the Wnt signaling, and the dorsoventral polarity is regulated by the BMP signaling. Yeah, so if you, for example, activate the Wnt signaling, you can achieve two-headed, for example, or multi-headed planaria flatworm. And similarly, yeah, it's, I think. Yeah. So let's move on. So we will discuss a little bit more about this blastema now. So now we go a little bit higher in the vertebrates, and we'll consider the example of this salamander, and we'll see how it regenerates its limb. So the first thing, if you make a cut on the limb of this amphibian and this salamander, the first thing that is formed is that it's the apical epidermal cap. And so it is immediately, for example, you can see by this, it is immediately covered by this green, and it is shown by green here. It is immediately covered by this skin layer. So the wound is covered by this. If you make a cut, if you make a wound and cut out the part of the limb of this salamander, the first thing that happens is that the wound is immediately covered by an epidermal cap. And under this cap, there is a blastema that forms. And this blastema is a mass of undifferentiated cells, and it is derived locally from the nearby connective tissue. And this blastema is very similar to the blastema that is formed in planaria in the flatworm, where the neoblast cells, they start to proliferate. While in case of amphibians, the blastema is composed of the connective tissue, mainly the connective tissue cells. It can also be, it can also contain muscle cells. Yeah, so the blastema is important thing is that the blastema cells, they are limited in their, or restricted in their developmental potential, and it is always their differentiation is always into a related cell type. So a connective tissue, for example, cells will only, or for example, if the blastema is formed by the muscle cells, it will always give rise to muscle cells. And that is nicely, for example, demonstrated by this experiment. So what they did is they make transgenic animals. So transgenic animal means that they change one of the gene in this salamander and make it fluorescently labeled. So they change the genome and then they add a fluorescent marker next to a gene so that when the gene is expressed, its protein product is coupled with a green fluorophore or a red fluorophore, for example, GFP or RFP or mCherry. And in this case, for example, they coupled, for example, a muscle gene with a green fluorescent protein so that all the muscles of this transgenic animal, they are green in color or they fluoresce. And so then they made a cut at this position into this muscle and into the limb, and then they follow what happens. So what happened later on was that the connective tissue cells that form the blastema, they are sorry, the connective tissue cells that form the blastema, they are mainly from the muscle cells that were de-differentiated, and then they grew along because the part that was regenerated, it turned green. And so this also tells us that somehow these regenerating cells, even after de-differentiation, so first of course they need to de-differentiate, they lose the differentiation status, whether it was a muscle or whatever it was, but they still keep the memory of their differentiated state, and they again develop this green fluorescent muscle in the regenerated limb. Another thing that we understood from this experiment or from the studies in limb regeneration in amphibians is that the connective tissue actually guides the patterning. The connective tissue, it has information to guide the developing limb, and it somehow keeps this memory that there was what is more distal to the cut site. So proximal means behind the cut site, and distal means in front of the cut site. So what was in front of the cut site, it was gone, and now you need to regenerate, and this regeneration is dependent on the connective tissue, and connective tissue cells somehow retain this ability to know how to pattern the limb distal to the site of the cut. And yeah, so if muscle cells are used for the blastema formation, they can only give rise to muscle cells, which is shown by this experiment. But if general connective tissue cells, for example, fibroblasts or osteoblasts, they are used for blastema formation, then they can differentiate into slightly more number of cell types, like cartilage or connective tissue cells or muscle cells, can also make bones. So connective tissue has this ability to, it has a little bit more developmental, less restricted potential than muscle cells. So another important thing, the important consideration in the limb regeneration in amphibians is that the presence of nerves is very important for the regenerating limb. So what happens if you denervate the limb and then make a cut, then the blastema will still form, but it will not grow into the whole limb. So somehow then the nerves, and we will see how the nerves, they have this capability to guide the patterning and guide the regeneration process. And it was actually found out that the newt has this anterior gradient protein that is a nerve growth factor, and that is responsible for this patterning. Because when they added this nerve, the newt anterior gradient protein in the denervated limb, make a cut, then the regeneration happens normally. So this factor alone is enough for the regeneration, for the regenerating limb. But another interesting observation was that, so in case of a neurogenic limb, so what is a neurogenic limb? A neurogenic limb means that during the embryonic life, you remove the nerve, so the animal does not have nerves. And the interesting observation was that the a neurogenic limb, when it was cut, it grew normally. And that was a surprising observation, because we just have seen that the normal limb development is dependent on the nerve supply, on the nerves. And while in denervated limbs, they cannot grow unless they are provided with this nerve growth factor, the anterior gradient protein. And it was later found out that the anterior gradient protein is highly expressed in epidermis in developing limb, and it decreases when the nerves grow in. So in the case where the nerves are normally present, the cells or the connective tissue cells that grow or regenerate this limb, they are extremely dependent on this anterior gradient protein. But in case of the limbs or the animal which did not have any nerves present, they were not dependent on the anterior gradient protein, because they were normally not used to having this anterior gradient protein present. So that's why they grew normally. So this dependence on innervation on the nerve is basically imposed only after the nerves grow in. So before that, so this anterior growth factor was provided by the epidermis, and that's enough for the growth of the limb. So yeah, so going into a little bit more detail, now we will study how the patterning is achieved, and how does the limb knows which direction to grow, and what are the factors that regulate that. So the observation was that the limb regeneration was always in the distal direction. So for example, if you make a cut here, the limb will always grow into the digits. It will not grow back. And that's something that it's a memory that is built into the system. And we will see how. So then the scientists did a very interesting experiment, and what they did is they cut out in order to prove that the limb regeneration is always in the distal direction. What they did is they amputated the hand. So around the wrist region, they cut out the amphibian hand, and then joined this cut part into the belly. And once it's completely inserted and the wound is healed, they cut out the top part of the arm, so the humerus here. So they cut out, in the original animal, they cut out, the second cut was here. But now the limb is joined. So these radius and ulna bones, these forearm bones, they are now sutured into or completely inserted into the belly. And they make a new cut site here. And this is how it looks after the cut. And now what they observed was that the regeneration in both these sites was always in the distal site. So they both grew into arms and arm and digits. distal to the cut side. Although we already have the distal region here. So here, this, as you can see, this is the forearm region. Yet, when you make a cut here, it will grow into another forearm and another digits here. And this was actually very interesting. And the question was that how this positional information is encoded at molecular level. And this is also similar to this anteroposterior gradient that was that we discussed a little bit earlier in case of Hydra, that there is an inhibitory signal that, for example, the head inhibitory signal, it increases if you go down from head to the tail, and the tail inhibitory signal, it increases if you go up from tail to head. And similarly, so you have this PROD1, a cell surface protein that is expressed in a graded manner along the proximal distal axis in the limb. So this is proximal, this is distal. Proximal is towards the shoulder, distal is towards the digits. So this PROD1 is expressed in this graded manner, and it determines that it determines that it's, of course, its intensity increases or decreases. Let me see. PROD1. Yeah, so its intensity decreases as you go down from proximal to distal. So it's very high towards the distal end of the arm, and it goes down significantly. So that's why this is one of the reasons why, or this is one of the molecules, or one of the signals that determines that the growth is always in the distal direction. Because if you make a cut site here, there is a certain PROD1 expression levels in this region, and they are higher towards upside or towards the proximal side. So that's how the connective tissue blastema knows that it will not grow the tissues on the proximal side. Rather, it needs to grow the tissues on the distal side, where the intensity of PROD1 is lower. So PROD1, you can consider PROD1 as a kind of inhibitor of limb growth. So that, or inhibitor of proximal limb growth. And that's why in this both these situations, the limb grew always in the distal side. And PROD1, so this anterior gradient protein is the ligand for PROD1. So yeah. But there are instances where you can you can force to regrow the proximal structures as well. And that is actually provided by the studies of retinoic acid. And so the retinoic acid, it's a related compound to vitamin A, and it is also a nuclear receptor, and it binds to the genes directly, and increase or decrease their expression. So it's kind of a transcription factor. And so retinoic acid can change the proximal distal positional values. And this was shown by this remarkable experiment where they cut out the limb of the amphibian at the wrist, and then injected retinoic acid at the wound site. And what happened was that the retinoic acid not only grew the hand distal from the cut site, but it also grew all the structures until the shoulder blade. So it grew back the humerus, which is, you can see here as well. it also grew back this ulna radius, which are here. So this experiment showed that retinoic acid actually regulates the, or it can change the proximal distal positional values, and it's very important to regulate the levels of retinoic acid then. And PROD1 is a target of retinoic acid responsive gene. And so when its concentration is increased, the cells move more proximally. So the cells can generate structures more proximally. Okay. Right. Now the bigger question is why mammals cannot regenerate the limbs or cannot regenerate anything apart from the tips of their digits. So there are several molecular reasons. I don't know about what is the kind of more philosophical reason for that. But generally, mammals are more complex animals. They have more complex structures, and it's relatively more difficult to regenerate the organs in the mammals than in smaller animals. But still, just a newt or salamander is an exceptional case, and also the zebrafish. So let's consider this example, how mammals can regenerate the tips of their digits. So there is a connective tissue under the nail organ. So it's very important for the regeneration of the tips of the digits is to have a nail organ. So if you don't have a nail organ, then they will not grow. Or if you damage the nail organ, then the nails will not, the digits will not, tip of the digits will not grow. This nail organ has the connective tissue, which expresses this MSX1 transcription factor. And these are associated with, this transcription factor is associated with undifferentiated mesenchymal cells. And then BMP signaling is also required for digit tip regeneration, and it was shown experimentally that application of BMP to mouse digits more proximally can also elicit a regenerative response. But the important thing is that these mesenchymal cells or undifferentiated mesenchymal cells, they are lineage restricted cells, so they cannot grow into any organ at wish. They are kind of this progenitor cells, so they have a very limited ability to differentiate, so they can only differentiate into cells that can then grow these nails. But if you, for example, activate the Wnt signaling, it would promote the growth of nerves, and then it might lead to growth of the digits, or it might lead to growth of the limb in mice. Although, yeah, I mean, how the birds are doing radiation, how cells remember now. Okay, so yeah, so Mad asked that how the particular size of organ is achieved during regeneration and how cells remember or know where they have to achieve a particular size and not grow beyond that. So this is all dependent on the presence or absence of the growth factor. So like I said, that as the limb grows, for example, in case of this PROD1, as the limb grows, its concentration decreases. And at certain point in time, its concentration drops down. And that's, so for example, one of the signals that shows that now they have to stop. But of course, there are many unknown factors here that are may be playing a role, because it's not completely understood. And the reason why it's not completely understood is because humans cannot generally regenerate. And most of the research, you all know, that is going on in humans. And yeah, I don't think that the amount of research in at newt cells or newt or salamanders is equivalent to the amount of research going on in humans. So, and since humans do not regenerate, the amount of data or knowledge that we have in these lower eukaryotes is very limited. So far what we know is that there are these positional signals, and that help basically pattern the body, and there is a gradient of these signals, and this gradient actually determines when to stop. And of course there is also the nerve growth as well, and that also determines how long the limb will grow. But yeah, I mean, I have to say that it's not completely understood what is the signal to stop growing, and when to know that the complete patterning is achieved. But somehow it is encoded in the cells that that keep the memory of, like, for example, we saw that the cells from muscle cells, even though in the blastema, the muscle cells, even though they were de-differentiated and then re-differentiated, they still kept their memory of being a muscle cell. So they only gave rise to muscle cells. So similarly, there must be some genetically encoded messages that determine the patterning, but also some protein levels that determine how long or how big or how many digits, for example. So we have seen previously that if you, for example, activate Wnt signaling, I think we have seen it previously in one of the cases. So it was shown actually that if you activate the Wnt signaling in Hydra, you can it will grow multiple heads. So that will tell us that Wnt signaling is very important in knowing what structure to grow and how much to grow. And there again, it must have a gradient, some kind of gradient that goes down as you go from proximal to distal. But yeah, I mean, it's not completely known. And in the end, we will consider the example of heart regeneration in zebrafish. And that's also a remarkable capability. And the biggest question there was, again, what was the origin of cells that make the regenerative tissue? Is it the muscle stem cells, like the satellite cells that we saw previously that mammals have? Or it is the differentiated cells that first de-differentiated and then re-differentiated? So what was found out was that it was the mature muscle cells, the cardiomyocytes, that first they de-differentiated and then they started to divide. So, and this happened. So for example, this is the heart in Hydra, in zebrafish, and it has, so in humans, we have two ventricles and Hydra has only one ventricle. It's a little bit simpler, but the overall structure is the same. So it has these chambers that are lined interiorly with endothelial lining, and there is a pericardium, which also has two layers. What happens if you cut out, let's say 10-15% of the bottom of the apex of the heart? What happens in few hours is that there is, of course, clotting and endocardial activation. And endocardial actually activation, it starts expressing a particular factor called this RALH2. And yeah, so another thing, just before going into the details of signaling. So it was also shown that there is a transdifferentiation of atrial muscle cells. So this is the atrium, and the atrial muscle cells, they could also transdifferent. So they get them, somehow they get the message that there is a wound in the ventricle, and so there is an injury, and they start transdifferentiating into the ventricle muscles, and then they fill up this wound and uh, regenerate the muscle. So this yeah, so once there is a cut, is the cells in the, let's say, you can call it a blastema, it's a atypical blastema. But so at the wound site, the growth of cells or the regenerating cells, the source of the regenerating cells could be either the cardiomyocytes in the ventricle or the cardiomyocytes from the atrial muscles. And what happens is very quickly in 1 hour, so they start expressing this RALDH2, which is retinoic acid synthesizing enzyme. So we previously saw what retinoic acid can do. So it determines the proximal distal positional values, and it also kind of expresses in a gradient way. So this leads to activation of the signaling pathways that then so first the endocardium starts expressing. So endocardium is the inner layer, and then the epicardium, which is the inner layer of the pericardium. So the outer capsule of the heart, that is the pericardium, and it also has two layers. So the inner layer is called epicardium. So both endocardium, so endocardium is the inside layer of the wound, and the epicardium is the outside layer of the wound. And both of them, they start expressing this retinoic acid synthesizing enzyme. And that will basically lead to the proliferation of cells, and that will cover the wound. And also then the pericardium also covers from the outside, just like the apical cap in the blastema. And then you can see that so the first thing is this RALDH2. You can see from the green that is expressed both on inside and outside and at the wound side. But then also there is this GATA4 transcription factor that is activated in the pericardium. And this after this, the wound tissue, it starts vascularizing, and the blood vessels starts to develop inside this tissue, and then there is final electrical coupling of the tissue and dissolution of the clot. And this regeneration is dependent on complex signaling mechanisms. So first, you saw that there is a retinoic acid synthesizing enzyme that is expressed both in the inner and outer layers, which so retinoic acid then activates the sonic hedgehog and IGF-2 expression, which basically serve as the growth factor signaling machinery, and it leads to the growth of the undifferentiated cells, which then also differentiate into cardiomyocytes. And then within a week, there is this expression of the GATA4 that leads to the differentiation, basically. So the sonic hedgehog and IGF-2, it leads to the proliferation, and then GATA4, it leads to the differentiation of undifferentiated cells into cardiomyocytes. And then you have these more differentiation factors, these homodomain transcription factors, MSXB and MSXC. and they increase during regeneration, although they are not expressed during embryonic heart development. So it's a little bit different. So the heart regeneration in zebrafish is a little bit different than heart formation during embryogenesis. So I told you earlier that the adult mouse in the first 7 days of their postnatal life, they have this capability to regenerate their heart muscle. And although the muscle, the heart structure is a little bit different. So again, mouse is a mammal, so they also have 2 ventricles and 2 atria. And but still they have this, they kept this capability for first few days of life, and we will see why. And the genetic mapping experiments showed that the origin of cells in the adult mouse in the postnatal life and during the regeneration were also cardiomyocytes, which first de-differentiated and then they start to proliferate. So if you remember that it was, so we learned it during the muscle differentiation, was that it was very important for muscle cells to stop cycling in order for them to differentiate. So they have different pathways where they stop the DNA replication machinery, and in that way they could differentiate. And in order to de-differentiate the muscle cells, the first thing you need to do is to reactivate the DNA replication machinery or DNA cycling machinery. And that is the first step towards de-differentiation. Yeah. So similarly in zebrafish, there is this epicardial activation, which then activates, then acts as the apical cap around the wound site. And then there is inactivation of the hippo pathway. And basically, so hippo pathway basically regulates the formation of heart, and if it is activated, or if it is it is normally activated, and it prevents the growth of the tissue in the heart muscle. And if it is, so that's why the first step is to inactivate the hippo pathway. And so in the mouse neonatal heart, this is achieved by this large extracellular matrix proteoglycan, the agrin, and it basically inhibits the hippo pathway. So agrin has a role in neuromuscular junction in embryos, but agrin is also found in this early, its expression goes down significantly after the birth, but in those first 7 days, agrin is still present. And that's actually the main reason that agrin is a key molecule why the mouse cells, they can, or mouse cardiomyocytes, they can proliferate. So agrin binds to this cytoskeletal complex, the dystrophin glycoprotein complex, and that in turn leads to the inhibition of hippo pathway. And the YAP protein is released, which is normally bound to this dystrophin glycoprotein complex, and then it activates and such a cell cycle genes. So agrin is the key molecule, but after 7 days, the agrin expression or presence of agrin the agrin levels goes down drastically, and that's why the mouse also loses the ability to regenerate their tissue, muscle tissue. Another growth factor, transcription factor that is required for embryonic heart development is this MEIS1 or MICE1, and it blocks the cardiomyocytes from entering the cell cycle. So you need to block this MICE1 factor in order for cardiomyocytes to start again entering into the cell cycle and de-differentiate. And actually this so the postnatally the MICE1 expression increases, and this is together with the loss of expression or loss of the levels of agrin and increase in the MICE1 production. It prevents the cardiomyocytes from growing. So if you somehow manage to play with these two molecules, so increase agrin and decrease the MICE1 or inhibit MICE1 expression, then you can achieve, hypothetically, you can achieve the growth of the heart tissue. And this is an area where there's a lot of research going on, and this could be quite beneficial for the heart patients. And it would be interesting to know if humans have the same signaling pathways and molecules present. But as we have seen previously, in case of these embryonic stem cells, that mice stem cells and human stem cells, they are embryonic stem cells, they are different in their growth requirements. And for example, so the mouse embryonic stem cells, they require this leukemia inhibitory factor while for growth in the laboratory, while the human embryonic stem cells, they need this fibroblast growth factor. So, I mean, there could be differences, but at least this is a way forward, and a lot of research is actually happening in this area. So to summarize the regeneration, first, we have to admit that we do not know a lot how the regeneration is going on, but there are certain principles that we have learned from small eukaryotes or lower animals. Is that so there is a positional information that is encoded in the molecules in certain proteins, and that basically drives the growth process. Then the presence of nerves, of course, is important because it provides this growth factor that grow the nerves, and that it leads to then the nerves basically the lymph follow the nerves. And then you have this retinoic acid that is important for proximal distal positional values. You have Wnt signaling that is very important, BMP signaling that is very important for anteroposterior positional values. So all in all, so you need to have stem cells, sorry, not stem cells, you need to have connective tissue that is present, that has the capability to de-differentiate and then redifferentiate. So this is a little bit different from stem cell based growth, because in stem cells, they are already de-differentiated and they have the capability of self-renewal. While in case of regeneration, you have, in most of the cases that we have seen, you have these differentiated cell types that first de-differentiate and then they redifferentiate. And finally, in case of heart regeneration, you have this agrin molecule and the MICE1 that if you can manipulate it, you can lead to the regeneration of heart. All right, so that's all for today. If you have any questions, please go ahead. Okay. So, all right, so that's the last lecture, and then I think you all know when the exam is, and so good luck with the exam, and please write to me if you still have some questions. I can try to answer you as good as possible. All right. So see you. Allah Hafiz.